Hi all. So today's topic of discussion is salvage pathway of purines. Salvage means to rescue. So it is the rescuing or recycling of purines from nucleotide degradation. So salvage pathway is the recycling of purines from the degradation of nucleotides. So the starting material or the most important compound in this pathway is PRPP, that is phosphoribosyl pyrophosphate. PRPP is the most important compound in this pathway and it is the starting material of this pathway. Phosphoribosyl pyrophosphate, PRPP. Now the products of this reaction or this pathway are AMP, that is adenosine monophosphate, GMP, guanosine monophosphate, and IMP, that is inosine monophosphate. So AMP, GMP, and IMP. Adenosine monophosphate, guanosine monophosphate, and inosine monophosphate. So these are the products. Now the enzymes of this reaction are APRTase and HGPRTase. APRTase means adenine phosphoribosyl transferase. Adenine phosphoribosyl transferase. And HGPRTase means hypoxanthine phosphoribosyl transferase. Hypoxanthine phosphoribosyl transferase. Now the AMP, the product AMP is formed by the action of APRTase, adenine phosphoribosyl transferase. The products GMP and IMP are formed by the action of HGPRTase, that is hypoxanthine phosphoribosyl transferase. GMP and IMP are formed by the action of HGPRTase. Okay. Now a key point about this salvage pathway is in RBCs and brain cells. Salvage pathway is the only pathway for the production of purines because de novo synthesis does not take place in RBCs and brain cells. So only the salvage pathway takes place and it is the only source for purines in RBCs and brain cells. Okay, now coming into the reaction. It is a very simple reaction. Firstly, about the production or formation of AMP. Adenine react with PRPP, that is phosphoribosyl pyrophosphate, PRPP, in the presence of APRTase enzyme, adenine phosphoribosyl transferase, APRTase enzyme, to form AMP, and a byproduct of pyrophosphate is also formed. So adenine plus PRPP in the presence of APRTase, we get AMP and a byproduct of pyrophosphate. So this is the first reaction, the production of AMP. Now about the production of GMP and IMP. A single enzyme, that is HGPRTase, catalyzes the formation of both GMP and IMP. So guanine and hypoxanthine along with PRPP in the presence of HGPRTase gives us GMP and IMP respectively. That is, guanine gives us GMP and hypoxanthine gives us IMP. So guanine plus PRPP in presence of HGPRTase gives us GMP, and hypoxanthine in presence of HGPRTase we get IMP. And a byproduct of pyrophosphate, similar to the reaction formation of AMP, here also we get a pyrophosphate molecule as a byproduct. Now this guanine monophosphate and inosine monophosphate can be recycled back to guanine and hypoxanthine, that is by this reaction. That is, guanine monophosphate forms guanosine and then it is recycled back to guanine, and inosine monophosphate becomes xanthosine and it is recycled back to hypoxanthine. So that is not important for us. The important thing for us is this part. Okay, so once again, guanine plus PRPP in presence of HGPRTase we get GMP and PPI, that is pyrophosphate molecule, as a byproduct. And hypoxanthine in presence of hypoxanthine plus PRPP in presence of HGPRTase, we get inosine monophosphate IMP and a pyrophosphate molecule as byproduct. So this is the salvage pathway. Now clinical application. If this HGPRTase enzyme is deficient in our body, then Lesch-Nyhan syndrome is seen in that individual. So a detailed video about Lesch-Nyhan syndrome is already put in this channel. So you can refer to that for more information. In that all the details are clearly explained. So deficiency of HGPRTase, that is hypoxanthine guanine phosphoribosyl transferase, causes Lesch-Nyhan syndrome. So this is about the salvage pathway of purines. Now to summarize, it is the recycling of purines from nucleotide degradation. PRPP is the starting material. Products formed are AMP, GMP and IMP. The enzymes responsible are APRTase, HGPRTase. And in RBCs and brain cells, salvage pathway is the only source for purines. And these are the reactions. Adenine plus PRPP in presence of APRTase gives AMP. Guanine plus PRPP in presence of HGPRTase gives GMP. And hypoxanthine plus PRPP in presence of HGPRTase also gives IMP. So I hope that was clear. Thank you.